All right, we are at 1 minute past 2. I think we have all of our audio lines tied together. We have a slide pack, so let's have a webinar. Good afternoon and good morning to everyone. Just a few very brief announcements before I pass over to Frank. If anyone is joining us just on the phone and they want to download the slide pack, that has been uploaded. You can find it on the events section of the website. It's the top hit on there, and if you open up the event, you'll find a link to the slide pack on there. I must warn you now, it's quite a large file, so it will take a few minutes to download, depending on your bandwidth, but that's because Frank has given us a beautiful collection of images. The concept paper that this presentation is referring to is now on the front of the website and is open for commenting. So by all means, add your questions and comments into the chat window here, and probably more importantly, once the presentation is finished, please go and add your thoughts directly into the document, because that's of huge value to the authors. And then finally, this presentation is being recorded, including everything you say and type, and it will be available on the internet shortly, just so you're aware of that. Okay, Frank, over to you.
Very good. Well, welcome those who are joining us. I'm going to, so that I can actually see, I'm going to pop this on full screen, and you can see me leaning into my cell phone here, which will look ridiculous, but that's okay. Anyway, so welcome, people, to the, this may be one of the last, maybe the penultimate of these webinars that are related to creating the new astrobiology roadmap for the next several years. And what my duty is today is to report back to you on the work of a group who has put together a white paper. And the white paper's organizing question is, how can we best overcome our ignorance about microbial life on Earth? And I'm going to scroll down here. And there we go. All right, so our ignorance is in part related to technical limitations, and has been for much of human history, but it's also due to how we envision the structure of the tree of life. This is an artistic structure envisioned by Gustav Klimt. Here is another structure envisioned back in the mid-19th century by Ernst Haeckel, 1866. And as you can see, the place occupied by single-celled organisms in this tree of life is perhaps appropriately towards the roots, but the amount of space that is devoted to them is actually rather minor, and is in fact emphasizes the protists, the Monerans, the bacteria recognized at that time, being relegated to a minor place on the tree of life. So there are a number of special challenges that we encounter in microbiology that has made it difficult to, until somewhat recently, to properly place microbial life on the tree of life. One is, what is a species? What are the most phylogenetically informative characters by which we can establish the purported relationships among microbes? Importantly, and this is something that we dwelled on a good deal in this working group, is how do we overcome culture biases? And also, how do we connect various taxa with their ecological functions in the environment? And, are the functions that we can assay in the laboratory relevant to those functions that they perform in the external environment? And then of particular relevance to NASA, how do we recognize biogeochemical signatures of microbial activity? Well, in the early part of the 20th century, the foundations of microbiology were laid, by people like Koch and Winogrodsky and Bayerink. But for us, perhaps the most revolutionary figure in microbiology was Carl Woese, who recently passed away. And Dr. Woese, back a quarter of a century ago, envisioned a very different way of looking at the tree of life, a way that was based on nucleic acid sequences, which were just then becoming available. So here's a often reproduced figure from his seminal Microbiological Reviews paper in 1987, the universal phylogenetic tree determined from ribosomal RNA sequence comparisons. Well, this new view of life, which emphasized the information molecule of living organisms, or at least organisms here on this planet, really set the stage for revolutionary changes in microbiology. And these changes have only accelerated since the late 70s and 80s with the advent of whole genome sequencing that was catalyzed by the Human Genome Project and the acquisition of not only whole genome sequences that are enabling the construction of new phylogenetic trees that are based on whole genome phylogenies, but of course the production of metagenomes and metatranscriptomes directly from the environment. Okay, well, another special challenge that's faced by microbiologists in trying to organize how microbes fit on the tree of life, of course, is lateral gene transfer. Here's one of my favorite papers from PNAS about 5 years ago from Dagan et al., who constructed a series of trees connecting the vertical branches with the ancient, intermediate-aged, and recent lateral gene transfer events, coming up with this lovely figure C, which combines all of them. But this is clearly a fact of microbial life that needs to be taken into consideration as we're trying to overcome our vast ignorance. Well, it turns out, of course, that lateral gene transfer, not only within microbes, has had an important influence on the history of life, not just its phylogeny, but how microorganisms have been able to adapt and thrive in virtually every environment that we've looked, but also lateral gene transfer from microbes has played a huge role in eukaryotic evolution. Interesting paper from a year or so ago in Nature Communications summarizing the presumed lateral gene transfer events from bacteria and archaea into mosses and vascular land plants. Well, I think that we can assume that if there are life forms elsewhere in the universe, that these life forms likely shared molecules that encode information about how to make a living as well as how to make more living things, more replicants. And I'll just say as a side note that it's noteworthy that the possibility that life originated as a collective of such modules is in fact a major theme of the University of Illinois' Universal Biology NAI project. So the title of the paper that I would invite all of the attendees to examine and also pass the word to your colleagues is how can we overcome our ignorance about microbial life on Earth. We jokingly refer to this as the vast ignorance topic, and this topic was subsumed under what organisms remain undiscovered in our biosphere and what do they do ecologically. So, as is the case with the other white papers, we were charged with writing an explanation, a justification, and then a set of related subsidiary questions, which in the new roadmap will be molded into suggested programs for study for people applying for grants through ExoEvo as well as the NAI program. So the point that we sort of took off from is exemplified or illustrated on the right, where you have growth of biological databases, genomes, ongoing genome projects, and metagenomes in the various databases by year over the last 13 years, an explosion of data. But even with this explosion of data, our understanding of the tree of life structure and its functional output is incomplete. In many cases, particularly those cases related to metagenomic data, the structure and the presumed output is based on the RNA sequences, and the inferences that we can make about those RNA sequences relative to the physiology and ecology of cultured organisms and communities that we know something about in the natural world. The natural world now, including the microbiomes of the various macroorganisms that inhabit the planet. So the point here also is that we have been studying microbes like E. coli intensively for nearly a century, and yet recent studies have shown that we understand perhaps half, perhaps a bit more of what their genomes encode. This is obviously a problem as we're trying to relate the output of metagenomic analyses to making inferences about physiology and ecology. And the point is, and I'll illustrate this with a number of examples here in the next 2 or 3 slides, is that genomic analyses of each new environment that we look at shows unexplored diversity. The diversity doesn't seem to asymptote in many cases, and these analyses emphasize just the extent of our ignorance about microbial life on the planet. So, as far as NASA is concerned, an increased knowledge of genomic diversity and a better understanding of the physiology and ecology of microbes on the planet should reveal new ways to envision how life might have emerged and how we could detect life on other planets. So this emphasis on dealing with the vast ignorance problem has been a concern of the major funding agencies that at least I and most of you guys have dealt with over the last 20 years. No doubt you're aware of the NSF MIPS, Microbial Observatories program. The U.S. Department of Energy has pumped a lot of money into microbiology over the last 20 years in the fields especially of bioremediation and bioenergy production. And, of course, the NIH with the human microbiome projects have supported this challenge of overcoming our vast ignorance over the last decade or so. As far as NASA is concerned, and specifically the astrobiology program, meeting this challenge of overcoming the ignorance has extremely important ramifications. First of all, microbes inhabit the most extreme environments on Earth. We've known for a quarter of a century or more about the marvelous vent communities and their associated microorganisms that are the primary producers there. But virtually everywhere we've looked, and here I'm showing some examples of projects that are whose publications have been within the last 18 months or so, publications by NASA-funded researchers where microbial life has been found in ice, like Vita in Antarctica, interesting new interactions between bacteriophage and sulfolobus species studied by Rachel Whitaker and her colleagues in Yellowstone, and also other such extreme environments, and another example here of microbes in basaltic glasses in the Guaymas Basin. So the assumption that we as NASA investigators carry into this challenge is that by an increased understanding of the most extreme environments on Earth, we're liable to get a better understanding of what we might expect in terms of microbial life on other worlds in our solar system, and also in investigating microbes in these extreme environments, we can get some inference into the kind of biogeochemical signatures that they leave, which, if that microbial life no longer existed, could at least provide evidence that it once did. Well, microbes also drive the biogeochemistry of the planet. All of the energy converting processes and processes involving the transformation of various metals and metalloids are primarily catalyzed by microorganisms. These two figures that you see before you now were taken from a figure by Falkowski, Fenchel, and DeLong, published a number of years ago in Science. Paul was a member of the Roadmap team and contributed to our discussions, emphasizing the importance of microbial biogeochemical transformations. And the assumption is that because this is the modus operandi for our planet, if life exists elsewhere, microorganisms are likely to be essentially involved in those processes elsewhere. Well, the challenge of overcoming our ignorance is just now being met, in part because of the new tools that we have available to us for metagenomic and metatranscriptomic analysis, but also a lot of new tools in terms of looking at biogeochemical signatures of microbial life in natural environments. And these studies are showing us some interesting surprises. The amount of work that goes into these papers that I'll show you in the next 3 slides is enormous. But the explanation for these patterns that are emerging from these studies is still a long way from us. For example, here's a paper by Shearer et al. In 2012, PNAS is a comprehensive community and biome structure function study. There are some interesting and unexpected differences in alpha diversity and in terms of metagenomic richness. Perhaps not unexpected, cold desert microbiomes have lower richness than, say, temperate deciduous forests. But there's a lot of spread in these data, and until we have more sample points in all these different types of environments, it will be difficult to make conclusive statements about the effect of environments on microbial diversity. Also interesting patterns observed in the number of reads. This is percent of reads. These are different categories of genes predicted from the sequences obtained from these different environments, now simply broken into 2 categories, desert and non-desert soils. And you see that in some cases, for example, in protein metabolism, you have a greater percent of reads. This is not the absolute number, but a greater percent in desert soils. This relationship is significantly reversed in non-desert soils with respect to virulence, disease, and defense. So I would argue that these powerful studies are our first pictures of, first really detailed pictures of communities' natural histories. Another recent paper by Caparaso et al. Looking at the metagenomic data from the Western English Channel emphasizes a couple of points that we talked about over and over again in our discussions. If you look at A, you see these seasonal patterns in terms of, here we have by date from January red down to December in blue, and predictable patterns in PCO unweighted unifrac. Here you have phylogenetic diversity plotted against sequences per sample, and what I mentioned earlier that we in these studies often don't see an asymptote is indicative of just how much we still have to learn. And then in panel C, the largest fraction by far in terms of relative abundance of reads is in unclassified bacteria. And so these reads do not provide sufficient data, or did not provide sufficient data to these workers to make a call as to their phylogenetic affinity. And then the last slide I wanted to show you is that there are people now who are using the relatively limited number of global metagenomic surveys, using these data to make statistical predictions about changes in microbial diversity. This is a recent paper from ISME Journal looking at global patterns in marine bacterial diversity in different seasons across the globe, peaking at high latitudes in winter. Again, these are all patterns, but these are patterns that we could have not even guessed at even a decade ago. Another challenge, aside from the fact that just the bewildering array of different phylotypes out there, is connecting genomes and the elements of genomes to function. And we, of course, rely on well-annotated genomes of organisms that we have studied in pure culture for many years. But a point that I raised earlier that I would like to emphasize with this particular citation, this Global Functional Atlas of E. coli that was published 4 years ago in PLOS Biology, is that these workers at that time gave us the take-home message that about 30% of E. coli genes, arguably the best studied organism on the planet, remain orphans. And so our annotation efforts still have a long way to go. And of course, that annotation effort critically influences our ability to make predictions from metagenomic data. Okay, so there is some slides sort of backing up the explanation of the problem. And so the second task of each of the groups was to provide a justification. I think we've already done that to some extent. That life on other worlds is most likely to be microbial, and so we need better to understand our own microbiomes here on the planet in order to inform explorations elsewhere, and studies of extremophiles are, of course, extremely important. And then not just extremophiles, but certain members of the team pointed out that even in communities that are considered to be non-extreme, I mean, this is a very subjective call. What is extreme to us, what may be considered stressful to us may be a very comfortable situation, for other organisms, is that we need to consider the possibility that unknown organisms and undescribed physiologies can play significant roles in non-extreme environments. The conclusion to the justification section is that given the vast majority of microbes are yet to be cultured, we have to be missing enormous amounts of information about how they make a living, impact the environment, and leave biosignatures. So here then are a series of sub-questions, and the panel came up with 7 sub-questions, which could be the basis for future study. One sub-question is, how do different methods shape and reinforce our current views on microbiology and the tree of life? And in discussing this question, we noted that depending upon your technique, that you necessarily have an observational bias about the microbiology of your system, whether you're using pure culture versus mixed culture, the type of PCR amplifications that you are performing and their biases with respect to, what your primers are, what your targets are, biases that emerge from the analysis of metagenomic and metatranscriptomic data, and even observational biases when you are using multimodal approaches. The second question that we identified was, how can we identify the physiological characteristics of uncultured organisms? So there's this phrase out there, the great plate culture anomaly, which refers to the fact that we almost certainly have in pure culture a very tiny minority of all the microorganisms on the planet. And so it's very difficult to connect a single taxon that we discover and cannot culture to its function in the environment, and that problem is multiplied as we consider that the community in which that taxon is found. We referred a few minutes ago to one of the most important problems under question number 2, and that is annotation uncertainties and outright errors. And certain members of the group pointed out specific examples of annotation errors that simply get propagated and due to poor genome curation. Now, this emphasizes the importance of this task of genome curation and having this continuously reviewed and continuously updated, which is a daunting task considering how many genomes have now been sequenced and how difficult it is even to secure funding to do proper annotation updates on our model organism genomes like, E. coli and yeast and mycobacterium and so forth. An important activity which needs everyone's attention. Another challenge is that the taxa that we discover in so-called extreme habitats may have diverged widely from their nearest phylogenetic neighbors. So we get a read, we get an rDNA sequence, and we bend this organism with another organism that was found in a much more clement environment. The physiology of the newly discovered organism may have absolutely nothing to do with the physiology of the organism that is its nearest neighbor that was discovered earlier, simply because it was easier to get that organism from that environment. An important problem. We suggest that future metagenomic catalogs really need to be associated with data on the physicochemical environment. The last paper that I showed you guys, this recent ISME journal paper from last fall, where they did statistical predictions of microbial of microbiome dynamics on a global scale. One thing that they did do in that data was associate the metagenomic catalogs that they had with detailed oceanographic data. So that is a good example of perhaps the way that we should be going in the future. Another major challenge is an urgent need for innovative culture-based and non-culture-based analytical methods. I know that the NSF Microbial Observatories program had a major push towards improved culture-based methods of isolating organisms and also testing their physiologies, but a lot of progress still needs to be made on this front before we're able to answer number 2. Question number 3, a provocative question in my view, and one that I think that NASA scientists have potentially a lot to contribute, is can a fundamental physics and chemistry approach to defining habitable phase space act as a guide in seeing where our known tree of life is not populated as a guide to future exploration and research? So what does this mean? Well, the grand exploration strategy really harkens back to defining, if you will, for microbes in various environments, an n-dimensional niche space that is defined by physical and chemical properties of that environment, and then seeing who lives where. We have 3 dimensions here, but of course you could expand that to as many as you care. And then, after we do this, then spaces or volumes within this n-dimensional hyperspace, that is defined by these different ordinates, be it temperature, pH, oxygen concentration, etc., where we see holes. Those holes could be there because microbes have not found ways to adapt or to exploit those particular volumes. But it also could be that there has not been a focused enough effort to try to isolate organisms from those spaces defined by these different parameters. So an interesting idea which I hope the community will give us feedback on. The 4th question, how can some or many of the organisms we view as unculturable become culturable, or stated differently, what new approaches or modifications of current approaches can be used to enrich and ultimately isolate organisms currently known only by their DNA sequences? Referred to this in a number of different contexts through the presentation today. The point is that we are strongly biased in what we know, certainly in our ability to do genome annotations. That ability is critically dependent on our ability to get an organism in pure culture and study its physiology. Improved techniques for getting unculturable organisms into culture is essential to overcoming our ignorance about microbial life on Earth. A 5th question, how can we address the inherent difficulties and the biases of sampling remote and or extreme environments? The group pointed out that there are that the largest biome on Earth, for example, the marine subsurface, has been poorly sampled, polar regions as well, the continental subsurface, and that driven by the challenge to overcome our ignorance about microbial life on this planet, that NASA researchers could develop better tools for overcoming our ignorance about whether life exists or not on other worlds. Our ability to do remote sensing, our ability to do to capture samples for laboratory analysis from remote environments. This struck us as being an important NASA mission related to the broader NASA astrobiology mission. The next to the last question that the group came up with was, what are some of the undiscovered catabolic strategies that may be used by organisms in largely unexplored environments? Classic paper from back in the mid 2000s, the discovery of a new type of phototropism by Beha et al. A great paper pointed out the importance of not just looking for organisms, but looking for novel transformations is an important activity for NASA microbiologists. And here's just an illustration of an important and recent discovery, a paper from Miluka Fertelman et al. In Nature about 18 months ago, of anaerobic oxidation of methane, methane oxidation being coupled to sulfate reduction. And this leads nicely into the last question that the group came up with, and that is, can we develop methods to meaningfully tackle microbial communities as the entities of functionality and selection? So any microbial ecologist understands that microbes in nature certainly do not exist on their own as single organisms, but are often functioning, as one person put it elegantly, as members of consortia. And so, for example, down at in the lower left hand, you have an example of processes coupled between Geobacter and Thiobacillus. There are, scores, hundreds of examples of these sorts of coupled transformations where you have not only intimate physical association between organisms, but this association exists as a syntrophy to an extent that many organisms are hard to or impossible to tease apart, raising the question, what here is the organism? If, say, the energy extraction metabolism required to sustain both organisms, that one depends on the other, is this perhaps, it's certainly a unit of function, is it also a unit of selection? And we have next to no information or understanding about the population genetics of syntrophic interactions in nature, and even a very poor understanding of these population genetic processes in syntrophic interactions in the laboratory. So another area where we have vast ignorance about microbial life on the planet. Okay, it would seem that I have lost connection temporarily, so but that's just as well, because I was close to the end of my presentation.
Looks like you're back, so go ahead and...
Give me a second to get back on the horse. And let's see, do you guys see me? Yes or no?
We don't see you yet, Frank, but you can start your webcam again.
Okay, all right. So I apologize to everyone for the interruption, but this is pretty much the...
We're back. Thanks.
Okay, great. Thank you very much. All right, so we concluded by just with a laundry list of techniques and problems, isolating microbes, single-cell genome sequencing, just a laundry list of techniques that we thought would be of importance in addressing some of the questions that we came up with under our overriding questions, and some of the problems. Some of these are simply restatements of the questions that we've already addressed. So that's the conclusion of my remarks. I did want to add one thing, and that is that one of the virtual participants in this white paper development was Rob Knight, who was actually a co-author of at least one of the papers that I put up on the webinar today. And Rob wanted us to make sure that we made the community aware of a systematic effort through the Earth Microbiome Project to overcome our vast ignorance about microbial life on Earth. And so with respect then to Rob, here is the website, and I would invite all of you who are not familiar with the website to poke around in there. It's really quite inspiring and just illustrating both the nature of the challenge and the ingenuity of the people who are stepping up to meet that challenge. Okay, so I think that is all that I have to say, and that opens us up for discussion. And thank you very much for your attention, and that's it. Thank you.
Okay. Thank you, Frank. Thanks very much. For anyone who hasn't been to one of these webinars before, and I think we may have one or two new people, you have a choice. The phone lines are now open, so you can talk directly with Frank. And if it is easier, I see Paulie is coming in from Finland, and I know he often has some difficulties with the telecoms. If it's easier for you to type questions into the chat box, then you're more than welcome to do that as well. So, thoughts about how to overcome levels of ignorance?
Yes, and I would really like for someone to talk, because I'm tired of talking, and I would like to hear. I would love to hear from. I see that we have some distinguished folks with us. Ron Ormond, who's done some great microbial ecology. Any comments from you, Dr. Ormond? We have a typed message who says, I will translate. That's Ron says,
And while Ron is thinking deeply as he's typing, if anyone wants to use the phone lines, please feel free. Go right ahead. It's *6 to unmute.
Like an NPR begathon here.
Also,
waiting for questions here.
While you are all thinking deeply, let me just put your attention back to the link in the text box, the third link down, which will take you directly to the document that Frank and Co. have been working on. Now, if you haven't used Google Docs before, it's a very simple process. You just have to right-click on a particular piece of text that you'd like to comment on. You get a little equivalent of a Post-it note, and you can type your comments in there. We do ask you to log in with your Google account if you have one, just so it captures your name with your comment. If you don't have a Google account, if you could add your name to any comments that you write in there, just so the authors can get back in touch with you.
Well, there continues to be a typing message coming from coming from Ron.
Well, I'll chime in. This is Jen Glass from Georgia Tech. This is new to me. I haven't really been involved in the roadmap so far, but I am very impressed with what is online so far. I love all these questions. And one that I'd like some more information on is 3, this kind of fundamental physics and chemistry approach be used as a guide. So if we combine all the known parameters that affect organism survival and behavior, can we predict the types of habitats? Can you talk a bit more about this in particular, this question, and how this was formulated and kind of what you're envisioning for these types of approaches? Is this primarily modeling, or what kind of examples could we think of here?
I'm dimly. I'm trying to reach back to discussions that took place last summer. My recollection. It was an interesting mix of people that came to this roadmap meeting, and this particular idea, I am pretty sure. I'm not going to attribute it to a particular individual, but I think I know who the individual was. And this was a physicist, I would say, or an applied mathematician, but someone who has been involved in NASA projects, a number of projects as kind of a hired gun. And I thought it was a provocative idea. This was not my idea. And so the idea was to do. We've all seen, for example, EH/pH diagrams, and that's a very simple example. I have here this example of a three-dimensional space, but the idea is that have we missed certain organisms by the biases in our culture approaches? In other words, are there organisms that in fact inhabit that space, but because of our the limited number of possibilities in terms of constructing a new culture system, have we missed those? And so my take on this idea is to construct a hypervolume of as many important parameters as we can come up with, and then populate that. Not just for example, each individual dot here is not just a particular taxon, but let's imagine that the space that dot occupies defines the physiological tolerances of that organism with respect to those three parameters. This is an extreme example, but I hope you sort of understand the principle. If in fact, in fact there might be a populated region here. I don't know whether you all can see this or not. I'm drawing with the pointer. There's a populated region here, and so the person was suggesting that we find these spaces that seem to be bereft of microbial life and construct culture systems or seek out environments or seek out portions of environmental gradients where those regions are likely to occur, and to intensify our culture efforts to sort of bring out the microbes that may be living there. And conversely, if we find holes where we can neither culture nor can we find evidence of microbial life from non-culture-based methods, then the boundaries of that space, of that forbidden zone, is also interesting biologically. So it works both outside in and inside out. I don't know whether I've done justice to this person's idea, but that's my take on it. What do you think, Jen?
Yeah, that's really interesting. And I recently learned from a colleague of mine about this this kind of most wanted taxa list for the human microbiome, which I thought was pretty interesting. I wonder if we could kind of apply that as well for the environmental microbiome and some of these metabolisms that we predict are out there from thermodynamics that we haven't been able to get in culture yet. And this kind of relates to what Ron Ormland has posted here about not discarding the search for pure cultures and continuing to go after that, because in some cases I think we become so focused on the omics and all the cool omics approaches that are being developed, which we should be, but perhaps we're not always focusing as much time on trying to get some of these predicted metabolisms into pure culture. So,
absolutely.
But I love that question. That's a great one.
I mean, emphasizing what Ron said. I'll read it out. Oh, I guess everybody can read it. So, excuse me, Ron, I'm not dissing you. Everybody can see your question. But this not discarding the search for pure cultures is essential to the annotation problem. That is to say that so what? If we can take a stick of DNA and associate it, with a known organism from a very potentially isolated from a very different environment, it may tell us really very little about what that organism is doing in situ. Really, what we need is not only better curation of existing genome databases, not only more metagenomes, but also more organisms in pure culture whose genomes have been sequenced and whose physiological tolerances and unusual metabolisms are nicely described. So that, it begs interesting evolutionary questions. So let's say that you have organism A living in an extreme environment, most related to organism B that's living in a temperate or a clement one. What genes have been co-opted by one or the other to specific adaptations in their respective environments? I mean, that's an important sort of molecular evolution question that could really only be answered by studying the organisms in pure culture, or at least in their syntrophic interactions in the laboratory.
Hello? This is Ron. Can you hear me?
Yes, we can.
Thanks, Frank and Andy. I just wanted to make a comment that this could also go experimentally the other way, pointing out the work of Jed Furman, who for years was finding archaea in the deep blue sea, in fact, more abundant than bacteria, and couldn't make any sense of that because what we knew about archaea was they were extremophiles or methanogens, and those critters should not be living in the deep blue sea. And then it was several years went by until they finally realized these guys were nitrifying and outcompeting bacterial nitrifiers at the environmental concentrations of ammonia. And since then, any study on nitrification, I don't care whether it's in the deep sea or estuaries or lakes or sediments or soils, always has an archaeal component based on follow-up pure culture work. Anyway, how you doing, Frank?
I'm doing pretty good. I wish I could actually see you. How are things down in California?
It's very beautiful. The temperatures are lovely. We have blue skies and absolutely no water. We need water.
Yeah. No, I talked earlier today with a friend who just moved a faculty colleague who moved his parents from the Bay Area to Colorado, and he said driving over the mountains, he was absolutely shocked. There was no snow.
There's no skiing. I can live with that, but I can't live without water. So we're enjoying life on borrowed time.
Excellent. Good to hear your voice.
Okay, in the last 3 or 4 minutes that we have for this session, are there any other points that people would like to raise? And again, I do encourage you to add your comments directly into the document as well. We will be capturing anything that's been typed into the chat window here, but it's easier for the author team if you can also write your comments directly into the paper.
Great. And Jen, I will be coming down to give a talk at Georgia Tech sometime in the spring, probably in the early part of April, not on environmental microbiology, but by all means, check in, and I'd love to talk more about this stuff.
Oh, I see people just trying to get in before the end. Okay.
Great.
All right, I guess I was on mute. Thanks, Frank, and I can't wait to meet with you when you come. I'm sure we'll get more info on that from Lauren.
Yeah, excellent. I'll look forward to meeting you and to catching up with all you guys, okay?
All right. Thank you very much. Thank you, Frank. Thanks to our participants. The recording of this session will be up on the website. There'll be a direct link in the event entry for today. So if you want to share it with anyone else, just give us a couple of days to get it processed, and then you'll be able to point them in that direction. And with that, thank you very much, everyone. Cheerio. Bye.
Thank you so much. Thank you, Andy. Take care.